Good afternoon. We're going to talk about preparing for 100% testing. Are you ready? Regulations worldwide in pharma are now becoming quite stringent. Depending upon where you are in the world, you're subjected to 100% inspection. So Europe and some countries within South America are requiring 100% inspection. Canada has a little bit different guidelines where they require that each lot of raw material is tested and each container for the identity of its contents by a specifically discriminating identity test. In the U.S., we're not subjected to 100% identity testing, but instead what it is n+1, the square root of n+1 of each shipment of each lot of active and inactive components are required to be tested prior to release for use in drug manufacturing. So is 100% identity testing really possible? Well, typically the way it's done now is that samples come into the receiving bay, the sample specs are inspected, the container has to be relocated, the top of the container has to be taken off, and a sample removed, taken back to the lab for identification through the QA department. What's done in the QA department is they log into LIMS, enter the information, do the identity testing, determine whether or not it passes or fails based on the criteria of acceptance, then log back into LIMS to say yes it passed or no it didn't pass. A reviewer of the second nature has to go and clear the product for release. But what if we could usurp a lot of that sampling? Actually do sampling right at the point of entry. We wouldn't have to necessarily take the sample and send it back to the lab. There would be some real increased productivity realized and real time savings in addition to costs. This is the idea that we're getting at, is that instead of even opening the bag, can the analysis be done in the container through a plastic bag or even through a super sack? Now, there's certain criteria that have to make this successful, and that is, instead of bringing the sample to the point of analysis, we're actually bringing the analysis to the point of sampling. Whatever method we're going to use, it really needs to be easy to set up and very efficient to use, because typically people that are not trained in any kind of chromatography or spectroscopy technique are going to be doing the analysis. So we want to make sure that we can lock them out of the system, but yet increase 100% identity testing with great efficiency. So therefore, there's a real staggering business proposition for using anything, and what we're going to offer here today is a handheld technology. So not only are we doing 100% identity testing and making a faster turnaround time, what we're going to do is we're going to increase the quality, because we're going to propose 100% accuracy in terms of that identification. Not only that, but what it'll do is it'll minimize the risks of the out-of-spec production costs, because if we can ensure quality going in, we have complete control on the product coming out. The analysis efficiency is going to be done because we don't have to do the time-cost exercise of moving it into a clean room, suiting up, getting a grab sample, and taking it back to the lab. Not only that, but we don't have any consumables for doing the analysis at the point of sampling. And in some cases, we encounter a lot of customers where they have to send the samples off-site. So they're basically holding the product up until the third-party lab or contract lab can ensure that the quality is of the raw material is there. So sometimes we encounter customers that tell us, what do you mean we can do the analysis in a minute? There's a possibility for this. We wait 2 or 3 days for the analysis to come back. Our system can be put into LIMS, which is obviously very important, because then tighter inventory controls can be done. And not only that, but better facility management, because if you can isolate and identify whether the compound passes or fails at the point of inspection, it's easier to quarantine as opposed to waiting for the analysis time to come back from the lab. So here's kind of the model that would make 100% identity testing very efficient. So we have the quality assurance person opening the container and sampling and doing the analysis without sampling. If the identity matches, obviously it's accepted, gets the second review, and puts it off to the manufacturing facility. If not, the raw materials should be quarantined, and then follow-up analysis being done. What is it? Is it a possibility that we know what it is, or do we need to validate the idea that it is something else that shouldn't be coming into the facility? If the third-party testing or the QA lab does LC, GC, anything like that, and says, you know what, something was mistaken, it's acceptance. That should be the final criteria. If not, it should be rejected or sent back to the supplier. So at Thermo, what we've done is we've taken the very successful benchtop idea, which is in the quality control lab, and miniaturized it. So we have here, and I'll talk about these in a minute, our handheld spectrometers. We'll talk about the capabilities of each one, but what it does is it allows the same analysis that's being done in the QA, QC laboratory to be done at the point of entry. So our first system uses Raman spectroscopy. And Raman spectroscopy is great for numerous compounds, and you can see them listed here. APIs, organic solvents, sugars, starches, and many other compounds. In some cases, compounds fluoresce, such as silicates or brightly colored dyes, and they tend to fluoresce. So with the fluorescent compounds, what we do is we always look at the methods and the signatures that are acquired during sample acquisition and determine whether or not we have the confidence to be able to identify compounds that fluoresce. And obviously, things such as the dark plastic bags or water highly diluted solutions are not necessarily well suited for Raman spectroscopy. But to that end, we do have a separate answer, and that is NIR. NIR loves water, so anything that's a water solution can be analyzed by NIR. It's also great for APIs, organic solvents, and many other compounds. In addition, NIR doesn't care whether the compounds fluoresce or not. So if you have NIR capability and highly fluorescent compounds, quite often they're conducive to NIR analysis. What we've done is we've come up with a purpose-built solution which incorporate both Raman and NIR. And typically about 85% of the APIs and excipients and raw materials can be done with Raman spectroscopy. But every once in a while you have one of those outliers that fluoresce, or something that lends itself to NIR that tends to be NIR active and not Raman active. And so what we recommend is that you have both capabilities at your site. Now these instruments are not new. We initially launched these two types of products in 2007. We're on our second generation, and what they are is actually really meant to be designed for non-technical users. That means they can't get in there and mess with the database. They can't do anything except turn on and off the instrument. And you can see a couple of screen captures in the lower left-hand corner that really decide right there for the user whether it needs to be quarantined or if it gets passed off to the production area. And what's interesting about all of this is we can typically do an analysis in less than a minute. What they are is they're meant to be simple turnkey solutions. These instruments are provided with libraries on there, so a lot of the up and run time is very short. So we can actually go through packaging materials, and not only that, but should you have APIs or excipients that are proprietary, we can add those spectral signatures to the instruments so that you have your own proprietary library. They sit on the network as a database and then can be shared within sites. And obviously, instead of doing any kind of analysis, again, we can do the APIs and excipients should they be just for your facility. And here's how it works. So what we do is we take a spectrum, and the user doesn't see this, but what we do is we determine whether or not that measurement that we're acquiring matches with a certain level of confidence to our reference library. And if the uncertainty in that measurement is too great, what we do is we say that the sample fails. And if the uncertainty is less, then it passes. So all what is going on at the top is going on behind the scenes, but all the user sees is what's on the bottom, those two screen captures. Either the sample that you're acquiring either passes or fails. Now, if it fails, we can do a discover mode, meaning let's capture the spectrum and compare that to the laboratory results after the interrogation from the QC lab. Now, Thermo has a patented analysis on our TrueScan RM, and we call it PValue. We did not patent PValue, we patented the use of PValue, and what we have is the output of different types of phenol here with variations of the functional group. So if we're comparing phenol to the reference spectra of phenol, it passes with the 0.45 p-value. Now you'll notice with benzaldehyde, very similar to phenol, but we want to discriminate between all of the other possibilities of phenol with different hanging groups on there, such as benzoic acid, benzyl alcohol, and toluene. And these are the p-values that we get out of a 200-item library. Now, there's other handheld systems out there that say that they are using p-value, and I can tell you right now they're not. They may be using p-value, but not in the way the TrueScan RM is. What they say is that they'll be using a hit quality index, which is an actual match value. Match value is something that leads to a lot of false positives. If we do the same analysis with the typical algorithms that are out there called hit quality index, we see the following results. The criteria for passing is 0.95 and above. So if we do a phenol and compare it to a phenol spectra, it's going to pass. It should. It's identical. But with these four other compounds, we have HQI values, which are hit quality index, which are passing, but they're different compounds. So with this type of analysis, which is HQI, there's huge risk involved with false positives. So therefore, we're really proud of the fact that we do p-value the way we do, because we can discriminate these very similar compounds. So the question might be how to get started. Well, the first thing I always tell my customers is, how much money are you spending in the QC lab? Is there time delay? Would it be advisable and advantageous to bring the point of sampling and analysis to where you actually really want to do the identification? If so, sit down and figure out how much you're spending and how much time it's actually taking you to do this. Quite often, a purchase of one of our instruments, a return on investment can be realized in less than 6 months. If you're having trouble quantifying that, or you need some higher level reinforcement, we certainly can come and help you figure out those cost savings that can be realized. We encourage you to talk to other handheld vendors besides us. Certainly include Thermo in the mix. Ask them, how is your IQ/OQ PQ documentation? We have a 146-page document which allows for complete documentation and qualification of the instrument. What are their validation and software procedures? Are they closed? Are they open? Are they able to have someone go in there and change the criteria? That's in direct violation of a lot of regulations, and certainly in compliancy of 21 CFR Part 11 compliant. We have audit trails on our system. Are there peer-reviewed references? Do a demo with them. Take some challenge samples. Are some of your APIs or even your raw materials in their library? What we do with our systems is not only do we have the p-value, our patented algorithm for that, what we do is we have our qualification direct on-site, and we do it for you. We walk you through the procedure, we have it, we leave it with you, we sign it, so that if you ever get audited, the auditors will see the IQ/OQ PQ documentation, and you're all fine. We have numerous SOP templates, which are being leveraged at numerous pharmaceutical customers around here right now. We have instrument certification, we have statements of compliance, we champion and user training, and also we have a pipeline timeline discussion. Now here's the real thing. I told you to talk to other vendors, but what we've done is we've already gone out there and we're the market leader for this. So I want you to feel good about talking to Thermo, because we're at 24 of the top 25 global pharmaceutical companies. We're at 500 GMP sites installations worldwide and have over 1,000 instruments deployed doing raw material analysis as we speak. Our instruments are definitely within the compliance of all the pharmacopoeias listed here. And all of our instruments are being used at all of the regulatory enforcement agencies worldwide. We just found out that the evaluation at the bottom here with Rwanda, Ukraine, and Zambia has been successful, and they're currently purchasing instruments to really go after the anti-counterfeit drug market. So I encourage you to come over and see the handheld. If you don't want to walk all the way over to Building 9, I realize it's a long walk. Myself and my coworker are here. We've brought a TrueScan, and we'd be happy to show you the instrument. Thank you very much for your time, and we look forward to helping you become 100% compliable with ID testing.